Bismillah ar-Rahman ar-Rahim. Welcome again, guys. Welcome back to another video lecture by Kami, microbiologist. Today in this video lecture I am going to talk about B cell receptor in detail. In the previous video lecture, if you guys remember, we talked about the B cell biology, means the overview of the B cell biology. And in our view of the B cell biology, what we talked about the B cell structure, we talked about the B cell activation, we talked about the B cell development, we talked about the B cell differentiation, we talked about the B cell receptor as well, but in a short way, means to just clarify your concept about the B cell. So we already talked about each and everything, not in detail but just conceptually. So if you miss this video lecture, so here I will recommend you to go back to my channel and watch the B cell biology overview. Okay, so once you guys get idea about the B cell biology, so then the rest of the video which are regarding to B cell biology will be very easy for you. So without wasting any time, let's start the video. Okay guys, so now let me write B cell receptor. This BCR is also called as membrane bound antibody. So let me talk about a little bit idea about the B cell receptor. You know, B cell receptor is a kind of receptor which are found on the B cell surface. Okay, that's why we call that membrane bounded antibody. And this B cell receptor is actually similar to the antibody, but still it is different from the antibody. Now how different from the antibody? Because B cell receptor have the transmembrane domain, moreover they have a cytoplasmic domain. But on the other hand, secreted antibody don't have these things. And moreover, you know, this B cell receptor mainly activate the B cell. How? When the antigen are bind to the B cell receptor, so then it will provide signal to the nucleus of the B cell, due to which the B cell activated. And once it activated, then it will be differentiated into plasma cell, effector cell, and memory B cell. So this is importance of the B cell receptor. You know, with this B cell receptor, there are some other receptor molecules, accessory receptor, which we call it Ig beta, Ig alpha. You know, it's Ig beta, Ig alpha are also a kind of receptor which are associated with the B cell receptor, and it helps the B cell receptor to provide the signal properly to the nucleus. And these two receptor, means these two molecules, accessory molecules, are called as heterodimer of IgA and Ig alpha and Ig beta. Moreover, you know, this B cell receptor are almost, we have 50,000 different B cell receptor, and it means that it can identify more than 10,000 different types of bacterial or different types of microbial cells. Okay, so this is actually the specialty, the unique properties which are related to the B cell receptor. Now if I draw the B cell receptor here, so let's suppose this is the B cell. Okay, so inside the B cell what we have the nucleus. Inside the nucleus they have the genetic material, as you can see. So this is actually the B cell. You know, at outside of the B cell there are although different types of the receptors, but here I will draw two main receptors. Let me draw. These two are what? These two are actually B cell receptor. Okay. Remember guys, as we already talked, that B cell receptor are mainly, you know, formed inside this, means it mainly formed by this DNA. And the DNA are this DNA are transcribed into RNA. This RNA are then translated into protein. These proteins are then give rise to B cell receptors. Different types of the B cell receptor. Okay. Moreover, they mainly express two types of the receptor, which we call that IgD and IgM. means membrane bounded immunoglobulin delta, membrane bounded immunoglobulin mu. These two types of the receptors are mainly found on the B cell surface, and these are actually the BCR, means B cell receptor. Okay guys, so remember, these B cell receptors are mainly responsible to activate the B cell. Okay, now there are some other receptor molecules which are also present with this B cell. So let's suppose if I draw this B cell here, so let's suppose this is the surface of the B cell, as you can see. So let's suppose this is the B cell receptor, as you can see. This B cell receptor have transmembrane domain, as you can see. These black one are the trans domain, and moreover they have cytoplasmic domain, this blue one representing the cytoplasmic domain. This transmembrane domain actually hold the B cell receptor in the transmembrane, and this cytoplasmic domain actually hold the B cell receptor in the cytoplasm area. Both actually provided the signal to the nucleus of the B cell. So let's suppose this is the nucleus. With this B cell receptor, there are some other accessory protein molecules which are actually a glycoprotein, and these are also the receptors. Let me draw the first, and this is the second. This is the second. Okay, these two receptor molecule, this is actually B cell receptor, let's suppose, and these two molecules are called as Ig alpha and Ig beta. Okay, this combination is called as Ig alpha Ig beta heterodimer. It means that they are different from each other, and these two receptors are mainly bind with each other by a disulfide bond. This green one, let's suppose, representing that's disulfide bond, and moreover they have another long chain, as you can see, which are connected with each other, and it is connected to this nucleus. What actually these two heterodimers can do, they this B cell receptor, as you can see, the cytoplasm domain it is not near to this nucleus. So if it is not near to the nucleus, so we get a long gap, a very large gap, as you can see. So the distance between the nucleus and the cytoplasmic domain of the B cell receptor are very large, due to which the signal is very difficultly provided to this nucleus. So for the proper transferring of the signal, so what happened, they need this accessory molecule. First of all, this message is transferred to this chain of this heterodimer receptor. After that, then they will transfer the same message to this nucleus, and then the nucleus will show response. The nucleus will produce some kinds of responses against this kind of pathogen. So let's suppose this is a kind of pathogen which are recognized by the B cell receptor. So this is how the B cell receptor can perform their function in the B cell activation. Okay, so that's all about the B cell receptor and their main properties, and how it can different from the secreted antibody. I hope you guys get ready about the B cell receptor. Inshallah, in the next video lecture we are going to talk about the B cell development and their maturation in detail. So if you like this video, make sure to hit the like button, share this video, and definitely subscribe the channel. And if you have any kind of question in your mind, you can write the question in the comment. I will try my best to give you answer as soon as possible. So like this video, share this video. Thank you so much.